So we can begin. So don't forget, there's an exam on Wednesday at 5. And what we talk about today will be fair game for that exam. And then on Wednesday, you'll start a new section. Dr. Bauma will be here to discuss reproduction. So what we're going to do today is we're going to pick up where we left off on Wednesday before break. And you remember we were talking about the brush border. Before break was the brush border enzymes. Those enzymes were transmembrane proteins in the microvilli of individual enterocytes. And those transmembrane proteins were disaccharidases. They were the enzymes that would convert disaccharides. Could be maltose, could be sucrose, could be lactose rather. And those enzymes now converted those disaccharides into 2 hexosugars. And then there were transporters, sodium-driven co-transporters on the apical membranes of those epithelial cells. And those sodium-driven co-transporters could snag a sodium ion along with one of those hexosugars and move it across the apical membrane of the enterocytes, putting it inside the enterocyte. Well, we're going to finish that story today, but we're going to now emphasize the final digestion of proteins. Included among the brush border enzymes as transmembrane proteins are going to be these peptidases. They're called amino and carboxypeptidases. And as the name implies, what these peptidases do is they recognize the terminal amino acids, either at the amino terminus or the carboxy terminus. And they clip off one amino acid at a time. And by doing that now, they've liberated individual amino acids that could be snagged by the sodium-driven co-transporters and moved into that cell. So where before we were moving hexosugars, we're now moving individual amino acids. But of course, these enterocytes would fill up with amino acids if we simply let that run. And so there are facilitated transporters in the basolateral side, sort of parallels to GLUT2 we talked about last Wednesday. But now what we have are facilitated transporters for amino acids, and they're going to move the amino acids across the basolateral membrane. This will bring us round to what we're going to do with those hexosugars and those amino acids. And the fate of these molecules, is to go from the intestines to the liver. They're going to be put into the hepatic portal blood. And one of the early slides I'll show you today is an image of the villus. Now remember, the villus is a finger-like structure. It's a multicellular structure, contains thousands and thousands and thousands of enterocytes. And right against the basolateral membranes of all those enterocytes will be a capillary, an extension of the hepatic portal system, so that now we can strategically move those amino acids and hexosugars from the enterocyte into the extracellular space and into the blood, so that blood can sweep these nutrients away to the liver. Then we have to think about what's going on in the liver. The fundamental structural unit of the liver is called a liver lobule. It's a peculiar structure in that it's hexagonally shaped. But we'll see that at each of the vertices of the hexagonal shape, we're going to have 3 tubes. One tube is going to be a tube carrying the hepatic portal blood. A second tube is going to be a tube carrying blood from the hepatic artery. So if you think about this for a minute, you'll recognize that the blood from the hepatic portal system is going to be nutrient rich, but because it's passed through the small intestine, it's going to have most of its oxygen absorbed. So it's oxygen poor, but nutrient rich. The other side of that coin is the hepatic artery blood. It's oxygen rich. We need oxygen to feed the hepatocytes, which are going to be the cell type in the liver. And so we need the oxygenated blood to take care of those hepatocytes. But we also need to have hepatic portal blood, which is carrying the nutrients for the liver to do its job. If you look at the numbers here, it's about 70 or 75% of the blood coming to the liver is hepatic portal blood. 25 to 30% is oxygen-rich hepatic artery blood. This blood is now going to mix in a special kind of capillary in the liver called a sinusoid, and we'll talk a little bit about that. But the third tube that is part of this triumvirate is the bile duct. So where these, particularly the hepatic portal blood, is carrying nutrients destined for the hepatocytes, the hepatocytes also generate bile salts and bile acids, and those hepatocytes that generate, the bile salts and bile acids generated by the hepatocytes will be drained by the bile duct, and we'll talk a little bit about that. We're going to see that both glucose and amino acids are being taken up from the hepatic portal blood. The sinusoids are basically just a fancy form of a capillary. So they're formed by endothelial cells. And sitting right next to the endothelial cells will be the hepatocytes. Well, among the important proteins on hepatocytes will be GLUT2. GLUT2 is the bulk transporter for glucose. We ran into it when we talked about the basolateral membrane of the proximal convoluted tubule. We're going to run into it again today in the basolateral membrane of enterocytes. So we're now going to be able to move those hexosugars and the amino acids out of the hepatic portal blood and into the hepatocytes. Then once in the hepatocytes, the amino acids will be repolymerized into proteins. And the glucose will be repolymerized into something called glycogen. You may recall we referred to glycogen as animal starch. It's basically a polymer of glucose, these gigantic molecules. But in this case, the glycogen is highly branched. So we can store lots and lots of glucose in these highly branched structures of the glycogen. But the other function of the hepatocytes, in addition to taking up amino acids and hexosugars and reconverting them to their polymers, the other story here is that the hepatocytes are generating what is known as bile salts and bile acids. Bile salts and bile acids are a form of detergent. And the way we make the detergent is we take cholesterol. Remember, cholesterol is hydrophobic. And at one end of the cholesterol molecule, we link either a glycine or a taurine. Glycine and taurine are hydrophilic. So now we've generated this molecule which has a region that's hydrophobic and a region which is hydrophilic. You'll recognize that as an amphipathic molecule. It's effectively going to be a detergent. And so we're now going to use these bile salts and bile acids to emulsify triglycerides. Fats are an enormous component of our diet, but they're very difficult to handle in terms of digestion. So what we're going to do is we're going to bring them in. We're going to eat all this fat. We're going to bring it through the stomach. We're going to bring it into the small intestine. And then in the small intestine, one of the first things we're going to do is we're going to use bile to take these big globs of fat and convert them down into tiny droplets. The problem here is that the enzyme that's going to digest the fat, the pancreatic lipase, only works in an aqueous environment. And so if you imagine this gigantic beach ball of fat, but the only way we can convert this gigantic beach ball of fat into its component parts is to work in, sort of nibble along the edges. It's going to take forever to get to the center of that beach ball. So what we do is we use these molecules of bile salts and bile acids to take the beach ball and convert it into tiny little BBs. These are what they call fat droplets. It's all surface to volume ratio. And there's an enormous amount of volume to a small amount of surface area in the beach ball. But once you get to BBs, now you have the surface to volume ratio is much more favorable for the lipases to eat away at the edges and convert the triglycerides into one molecule of monoglyceride and two molecules. Once we've done that, we can now form something known as a micelle. You may recall the micelles are unstable. We talked about that early on in the course. But we can help stabilize these micelles made of monoglycerides and fatty acids by mixing them with bile salts and bile acids. Those micelles now can leave the bulk phase, enter the unstirred layer. And in the unstirred layer, those micelles with their molecules of monoglyceride and molecules of fatty acid, those molecules can now fuse with the phospholipid bilayers. They're extremely lipophilic. So the fatty acids and the monoglycerides will go right across the phospholipid bilayer and into the cell. But then we undergo this laborious process. We reconstruct the triglycerides. We take the monoglycerides and the fatty acids and put them into the endoplasmic reticulum and reassemble them into triglycerides. But we're not done at that point. We take the triglycerides, which we've made in the endoplasmic reticulum, move them to the Golgi apparatus. And in the Golgi apparatus, we mix these lipids, these triglycerides, with protein. That makes them a little bit more hydrophilic. Then coming out of the Golgi apparatus will be vesicles containing something known as chylomicrons. The vesicles are enormous, but each individual chylomicron may be more than a micrometer in diameter. They're just too big to get across the endothelial cells that are forming the capillaries of the hepatic portal system. So instead what we do is we allow these chylomicrons to move between the endothelial cells and lymphatic vessels. Those lymphatic vessels are called lacteal, and now the lipid goes in, the chylomicrons rather go into the lacteal and go through the lymphatic system and are dumped into the blood in the subclavian vein. If you have taken a sample of blood shortly after last Thanksgiving's big meal and waited an hour or two for digestion, then spun it down, and then looked at the plasma, you would see the plasma was actually cloudy. And the reason it was cloudy is the chylomicrons are so big they'll actually reflect light. And by reflecting the light, the chylomicrons, which have been put into the plasma, show the existence of lipid in the form of chylomicrons in the plasma. Now, we'll take those chylomicrons and convert them into other low-density lipoproteins, and then we have to go through a whole nother phase of breaking them down and reassembling them into cells. We're not going to go that far. We're just going to get them into the lymph. And finally, if there's time, we'll finish with a discussion of the endocrine pancreas. And I want to touch on this because this is an important aspect of all of our lives. All of us know someone who's a diabetic. And diabetes results because either there's a death of the beta cells in the endocrine pancreas. Either we kill those. Usually an autoimmune disease will kill these beta cells. In that case, we can't make any more insulin. But in other cases, there's type 2 diabetes, where we lose the ability of the receptor to function. So again, it's a classic story. We release a hormone, we need a receptor. The receptor is then necessary to allow glucose to enter muscle cells. That's the way we control blood glucose levels. It's by secreting insulin from the beta cells. The insulin binds to muscle cells, the insulin receptors, that produces a structure on the surface called GLUT4. That's a facilitated transporter for glucose. We can now move glucose into the cells. Without the insulin, without the insulin receptor, we can't properly move glucose from the blood vascular compartment into muscle cells. And the system breaks down in many people over a lifetime, either by destruction of the beta cells, so we can't make more insulin, or the destruction of the receptor for reasons we don't quite understand. But in either case, it doesn't matter whether it's a problem of the hormone or the problem of the receptor. Remember, the story is usually about the receptor. We have a difficulty in transporting glucose across the cells, and we'll spend a little time talking about that. So let's begin here with a slide. This slide shows the structure of the villus. Now remember, we ended with a description of the microvilli, and the microvilli were extensions of individual enterocytes. That's not what we're talking about here. What we're talking about here is a multicellular structure. These are the finger-like projections, extensions of the mucosa of the small intestine. These finger-like extensions are composed of thousands and thousands of individual enterocytes. So in this orientation, this is extracellular. So the unstirred layer would be right over here. Then this is the basolateral surface. So the apical surface faces the unstirred layer. The basolateral surface faces this internal region, which is adjacent to the hepatic portal vein and adjacent to this green structure called the lacteal. What we're going to talk about here in a few minutes is we're going to drill down and talk about what's going on in individual enterocytes. And where we left off on Wednesday, last Wednesday, was to take these, or Friday, we'll take these individual enterocytes and talk about the brush border enzymes. The ones we talked about last time were the disaccharidases. We're going to see there's a very similar story with peptidases. We'll walk our way through that. But in the end, what we're going to be doing is taking those glucose molecules or amino acid molecules, transporting them across the cytoplasm of the enterocytes and then releasing them from the basolateral surface. You'll notice there's a strategic orientation here. Those molecules now, the hexosugars and the amino acids rather, can move right into the hepatic portal system and be carried away to the liver where they're going to be dealt with. We'll find that lipids have to go through another process, but they'll be released as chylomicrons, and they'll go into this green structure, which is the lacteal. Lacteal is just a specialized term for a lymphatic vessel. So let's recall where we were when we left last Friday. And we were emphasizing the idea of the enterocyte. And we talked about the enterocyte as an individual cell, and the apical surface of the enterocyte was thrown into these folds. And we termed these folds microvilli. And when we lined up several of these enterocytes side by side, the microvilli in aggregate formed something known as the brush border. Now again, this will sit on the basal lamina, so this is the basilar side. And before we left, the molecules we were talking about as transmembrane proteins were disaccharidases. They converted disaccharides and came to the lateral side, facilitated transport. We're going to see very similarly here, in the apical membranes, there are amino and carboxy peptidases. The amino and carboxy peptidases convert peptides into individual amino acids. I'll remind you that out here, there's this unstirred layer. So this is the unstirred layer. A companion transmembrane protein in these apical microvilli will be a sodium-driven amino acid transporter. So of course this is going to snag an ion of sodium in the extracellular environment and a glucose, move them into the cytoplasm. And in the cytoplasm now, we're going to get glucose. I'm sorry, we will have put glucose in there. We're now going to have individual amino acids. And on the basolateral surface here, there's going to be a facilitated transporter. It's a facilitated amino acid transporter. And the amino acids now go through that facilitated transporter. And then along the basolateral side here will be capillaries. I'm going to show this as a capillary. This is the endothelium. And this is intended to be a capillary of the hepatic portal system. The hepatic capillary is carrying blood. It's carrying blood to the liver. So now the amino acids, and if we were telling the glucose story, it'd be the same thing. The amino acids and the glucose coming through that GLUT2 transporter are now in the blood. They're going to be swept away. So as long as there's a gradient here where we're generating individual amino acids in the extracellular space, we can put amino acids and glucose into the blood vascular compartment and sweep them away to the liver. And that's where we're headed now. We're going to carry this blood to the liver. Over here, talk about blood to the liver. In the hepatic portal system. You can read here blood. Well, to understand what's happening to this blood, we have to spend a little bit of time on something known as the liver lobule. The liver lobule is said to be the fundamental structural and functional unit of the liver. And the liver lobule is a hexagonal structure. I'll show the 6 sides. Now, at each one of the vertices, there's going to be 3 tubes. The first tube I'm going to show you here is a tube of the hepatic artery. And the hepatic artery is carrying O2-rich blood. We'll see in a moment that there are going to be thousands and thousands of hepatocytes in here, and they need oxygen. So that's coming from the hepatic artery. But then there's a companion vessel here, right alongside, which is an extension of the hepatic portal vein. The hepatic portal vein is, of course, carrying nutrient-rich and O2 poor blood. So at each one of these corners, we have a repetition of this pattern. And its companion vessel from the hepatic artery. Those feed into, and I'll show this in green, these feed into a specialized set of capillaries. These capillaries have a name. These are called the liver sinusoids. But there's nothing fancy about them. They're just capillaries composed of endothelial cells. Then right next door to those capillaries, right next door to those endothelial cells, are stacks and stacks of these cells here, I'm showing in orange, which are called the hepatocytes. So at this point, we now are mixing the blood from the hepatic portal system and the oxygen-rich blood from the hepatic artery in the sinusoids. If you want numbers to this, it's about 30% of the blood coming in is from the hepatic artery, maybe 25, and about 70 to 75% is coming from the hepatic portal system. And then we're mixing that oxygenated and nutrient-rich blood here in the sinusoids. And then all of that blood drains through this region here, called the central the central vein drains the liver lobule. So that's the overall structure. We have these next door to the hepatocytes. Now in a moment we're going to put which is part of the bile system, but let's concentrate on this right now. The next thing I'm going to draw is I'm going to imagine that we can enlarge this, and we can imagine where the hepatocytes are sitting next door to the endothelial cells of the sinusoids. So we use our sort of typical shorthand here. These ovals are meant to be the endothelial cells. Label this as sinusoid epithelial cells, not epi, endothelial cells. And then right adjacent to those endothelial cells will be these orange structures, which are the hepatocytes. So these are meant to be the hepatocytes. Now remember, much of this blood in here has come from the hepatic portal system. So it's rich in glucose. And it's also rich in amino acids. So let's deal with the glucose first. These hepatocytes, along the membrane facing the endothelial cells, have transmembrane proteins that we've run into before. These are called GLUT2. And you recall GLUT2 is the bulk glucose transporter. We ran into this glucose transporter in the basolateral surfaces of the epithelial cells of Bowman's capsule. It's the same story here. It's in fact the same protein. And now what we're doing, and we also ran into them in the basolateral surfaces of the enterocytes. So wherever you see these high concentrations of glucose, we can move glucose across membranes simply by diffusion by using GLUT2. Well, the GLUT2 here allows the glucose to leave the blood vascular compartment and go into the hepatocyte. Once in the hepatocyte, the glucose is reassembled into glycogen. Of course, glycogen is a polymer of glucose. So we're taking these glucose molecules, reassembling them with 1,4 and 1,6 linkages. We're making a gigantic branch structure. We're storing glucose in this way. So we can store glucose in these hepatocytes as glycogen. Similarly, there will be facilitated transporters for amino acids. And these amino acids now can leave the blood vascular compartment in the hepatic portal system and be taken up into the hepatocytes. And the hepatocytes will now assemble, a better word is polymerize, amino acids into protein. So we're taking these individual amino acids and reconstructing them into gigantic proteins. Most of these proteins in the liver will be albumin. So these hepatocytes, these liver cells, are busy cells taking up the remnants of our digestive system, the hexosugars or the amino acids, and reassembling them. But they also do something else. At these vertices, there's a third tube. And this third tube, and there's one at each point, these are called canaliculi. And they carry bile salts and bile acids, which are synthesized by hepatocytes. So these hepatocytes have a number of jobs. The first job that we've talked about here is how they reassemble glucose into glycogen. The second job is how they take amino acids and reassemble them into protein. The third job is going to be making bile salts and bile acids. But if you take a toxicology course, the other thing that these liver cells do is they detoxify any number of nasty molecules we may take in with our diet. And so they're detoxifying any number of other molecules, and they have whole enzymatic pathways in these cells to do that. We're not even going to touch on that. So these are really busy and important cells. So let's see what happens when we use these hepatocytes to make bile. Now the bile doesn't go into the blood, it goes into this specialized series of tubes. So let's talk a little bit about bile salts and bile acids. Now you remember probably when we're talking about the digestive system that we talked about, or actually you may remember when we talked about the cardiovascular system, we said that when blood is broken down, we have these bilirubin and biliverdin, these bile structures, but they're a waste product. These are actually functional. They're a different molecule altogether. Where biliverdin and bilirubin were the result of breaking down porphyrin rings as best we could, these are basically detergents. So let's take a minute and look at how they're detergents. We can say that they are amphipathic molecules. And when we look at them, we'll recognize immediately the structure of cholesterol. These ring structures, I wish I'd made this up. And out here, on this 5-membered ring, we put a molecule either of glycine or taurine. Glycine and taurine are charged or polar. This region here, the cholesterol, this is hydrophobic. So taken together with this hydrophilic point out here, combined with a hydrophobic region in here, we have a molecule that's amphipathic and serves as a kind of detergent to emulsify fat. So again, the system we're talking about has a component which is blood vascular. These capillaries contain a mixture of oxygen-rich blood and nutrient-rich blood. We'll use that blood to make glycogen from glucose and make proteins from amino acids. But the other function here is to make bile. And we make the bile and we put it in molecularly. Let me show you a little bit of anatomy here, see if we can get us all on the same page. So this structure is a pretty picture of what I drew in 2 dimensions. And it covers everything that we talked about. You'll notice that each one of the 6 vertices, there are these 3 tubes. One tube is an extension of the hepatic portal system, one tube is an extension of the hepatic artery, and the 3rd tube is extension of the bile system. The blood is delivered from both the hepatic artery and the hepatic portal system into these lavender or purple structures called the sinusoids. And the sinusoids now are aligned right next to them with these thousands and thousands of hepatocytes. Then all this blood, which has come through the hepatocytes, now empties into the central vein and is drained out of the liver into the systemic vasculature. And we'll see a little later that that's where we're going to get high levels of glucose after a big meal. Now this is a close-up of the same area. Again, the hepatic portal vein, hepatic artery, feeding blood into the sinusoids. And the sinusoids, the blood is now flowing through and emptying into the central vein. And then this structure here is the 3rd tube. This is the tube which is part of the bile system. And we're going to be generating this bile salts and bile acids by the hepatocytes, dumping them into this structure. And those now, eventually, in the liver, from the liver, will empty into this structure. This structure up here is the gallbladder. It's simply a storage device. It's a storage device where we generate, where the bile salts and bile acids generated by the hepatocytes are stored in the structure. You may remember the term cholecystokinin, bile sac move. Well, cholecystokinin is that peptide hormone that we talked about that would bind to the acinar cells. But the peptide hormone, in this case, binds to smooth muscle on the gallbladder, and in so doing causes the gallbladder to contract and squeeze bile into the small intestine. And the reason we do that is we're going to use that bile in the small intestine to emulsify lipids. Do it this way. Here's the structure of the bile salts and bile acids. This is the cholesterol. And out here on this carbon of this 5-membered ring, we can either add glycine or, or taurine. The main idea here is that these are charged, in this case, or polar. And because of that, this molecule, end of the molecule interacts with water. This molecule interacts with fat. By using those molecules, we can now take these gigantic globules of fat and convert them into small fat droplets. That's what this figure is trying to show. It's trying to show we have this great big fat globules come in with one of our meals. You can think of this as sort of a beach ball of lipid. And because there's such an enormous volume here, there's not a whole lot of surface area. And the enzyme we're going to use to break down the lipids is called lipase. Only works in an aqueous environment. So the only way we can digest these lipids is to work from the edges. So one of the first things we do is we take the bile salts and mix them with these fat globules and break these fat globules up into these droplets. With the droplets now, we have a much greater surface to volume ratio, so we now have more surface to attack by the lipase. And that's where we're headed now. We're going to think about how we can attack those lipase molecules and what the fate of those molecules will be once they're broken down. So again, we'll go back to this image. Remember that the pancreatic lipase is working in the bulk phase. And what's going to happen is we're going to get these BBs, effectively, which we're going to now label as micelles. Now, micelles are composed of monoglycerides and fatty acid, emulsified Once these enter the unstirred layer, because they contain monoglycerides and fatty acids, once these bump up against the phospholipid bilayer, the monoglycerides and fatty acids are so hydrophobic, they're so lipid soluble that they go right into the cell. So in the cell now, we're going to get monoglycerides and fatty acids. We're not done yet, because the next fate for these is for those molecules to enter structures that we talked about a long time ago, called the endoplasmic reticulum. And in the endoplasmic reticulum, the triglycerides are reassembled. The monoglycerides and fatty acids are reassembled into triglycerides. But again, triglycerides are pretty insoluble, so they're hard to handle. So what we do is we break off vesicles here, and the vesicle now joins the Golgi apparatus. The vesicle joins the Golgi apparatus and deposits the triglycerides. And then go back up above. And in the Golgi apparatus, the triglycerides are mixed or complexed with proteins. And so in the Golgi apparatus, we're going to generate these structures called chylomicrons. So we can spell that out in words and we'll draw it. In the Golgi, the triglycerides that were reassembled in the endoplasmic reticulum are complexed or mixed with protein. To form chylomicrons. And these chylomicrons, which I'm going to show as red dots, in the Golgi apparatus are assembled into a vesicle. So this is a vesicle from the Golgi containing chylomicrons. Then those chylomicrons are brought in the vesicles to the basolateral surface with their chylomicrons. When the vesicle fuses, as we've seen by exocytosis 1,000 times in here, then the chylomicrons are released from the basolateral surface. But these chylomicrons are too big to enter the capillaries. They can't get into the blood capillaries. So instead, the fate of the chylomicrons, enter the lymphatic system. Through lymphatic capillaries. Called lacteals. This lymph then carries the chylomicrons, as with other lymph, is dumped back into the blood vascular system in the subclavian vein. So that's basically how we cycle the lipids. Now, I'm not going to get into the story of the fate of the lipid once it gets back into the blood vascular compartment. There's a whole nother story about how we tear it down again and rebuild it. We're not going to go down that road. But this is basically how we have to deal with lipids. Lipids are a real pain because they're so water insoluble. So we go to these great lengths to digest lipids. And the reason we do that is because there's so much caloric energy in every triglyceride molecule. All those acetates in the fatty acid side chains can be plugged into Krebs cycle to generate lots of energy. So that's why we do it. The last couple minutes, let me finish the story kind of quickly with something I think is really important. And we're going to start here with a structure we're familiar with. This is meant to be the exocrine pancreas. It's the crenellated structure of the exocrine pancreas. It empties into the small intestine. Then beyond this region of the exocrine pancreas, there's another region of the pancreas, which we call the endocrine pancreas. And there are regions within the endocrine pancreas which are called islets. And these islets contain a special set of cells called beta cells. And the function of the beta cells is to secrete insulin. What happens if we look at these beta cells is one of the major proteins on the beta cell is, again, an old friend, which is GLUT2. So what's happening here is that if we go back to this story, where the blood is being carried to the liver, the blood flows through the sinusoids. But whenever there's, after a big meal, there's excess glucose. The glucose now flows out into the central vein and out into the systemic vasculature. When that blood comes back and reaches the endocrine pancreas, that glucose enters the beta cell. This is the beta cell. It enters the beta cell and signals the beta cell to secrete insulin. Now, all of our muscle cells, put a muscle cell up here, contain on their surfaces insulin receptors. So this is meant to be an insulin receptor. So in the face of a high glucose meal, so you've just had a bunch of spaghetti, and now the glucose levels in your blood go way up. The response of the beta cells is to recognize that glucose and secrete insulin. The insulin then comes out and binds to the insulin receptor. And in the process, this starts a cascade of events. So we get exocytosis of GLUT4 containing vesicles. GLUT4 is another form of glucose transporter. So now it can take the glucose in the blood and move it into the muscle cells. That's how we get glucose into the blood. I'm sorry, that's how we get glucose into cells. And this is the basically result of the story I told you earlier of the Russian scientists who were removing the pancreases from dogs. What had happened is they removed the pancreas. When they removed the pancreas, they removed the beta cells. When they removed the beta cells, the dogs couldn't respond to glucose in their diet. So the blood glucose levels went way, way up because there was no way to get the glucose in the blood into these cells. Let me finish this with something that's important to all of us. And that is, diabetes is a big problem. But there are two forms of diabetes. One form is where we destroy the beta cells. It's probably an autoimmune disease. Our immune system sees those cells as foreign, and we destroy them. We can't make insulin anymore. There was a story yesterday, just I think it was in the Washington Post. Someone has developed a series of stem cells which contain the beta cells. They've basically made beta cells. And they've injected them into this one person, and that person was diabetic, and he's recovered from his diabetes. We'll see if it holds. The final piece here is to say that the other way you get diabetes is the receptors. That's type 2 diabetes. So type 1, we lose beta cells, we lose the receptors. And as we've said so many times in here, it's the loss of the receptor that's going to be important. Well, thank you. It's been a great semester. This is my last lecture. Dr. Baum will be here on Wednesday to talk about the reproduction system.